Welcome to Study Loop.
For this question, we're looking at the histone proteins of DNA and either the methylation state that they can exist in or the acetylated state. So the function of histone proteins is to help bind and condense the DNA inside the cell. So you can imagine that we have much DNA and it can be difficult to contain it inside one type of cell. So to manage that, we're going to supercoil it and condense it together. However, we also want to be able to read certain parts of the DNA when we want to, say, replicate it or to produce certain proteins from the DNA structure. So to do that, we have to unravel certain portions so that they can be read. And histones play a large role in this process. So in these two forms, in the methylated form, our histone proteins are going to be closely associated together, whereas the acetylated form, these are going to be more distant and open. And these just differ by what type of group is attached to each of these proteins. So in the methylated form, you're going to have the methyl group, whereas in the acetylated form, you'll have an acetyl group instead. And in each of these states, you're going to have the DNA tightly wound around these protein molecules, typically through the interaction of intermolecular forces, such as polar to nonpolar interactions, or say covalent bonding. The difference between these two is that in the methylated form, these are going to be more tightly wound around the proteins, and you're going to have less space in between each of the histone proteins. Whereas in the acetylated version, these are less tightly wound and you have large open areas. This is important because inside these large open areas, you're going to have the gene that needs to be transcribed and translated. And there's enough space here to allow for the attachment of the polymerase molecules and transcription factors in order to read and transcribe the gene to produce your mRNA product. Whereas in the methylated group, some genes may be hidden inside the tight coiling, or there may not be enough room for the polymerase and transcription factors to attach. So in this form, you have the decreased amount of transcription, whereas in the acetylated form, promotes the transcription from occurring. So here, if we're looking for a cancer where we want to keep genes on, per se, or activated, which could be from preventing certain tumor prevention systems, such as P53, from being turned off. Basically, we want the cell to monitor itself for cancer and to signal if something's wrong. We want to maintain this acetylated state and prevent conversion into this methylated state where those protective features would be turned off. So here, the best answer to this question is going to be A, that these drugs that want to protect these features of the cell need to prevent the removal of these acetyl groups, and we want to prevent the attachment of these methyl groups to our histone proteins. So here you should choose answer choice A.